was nice. This is very helpful. Then we can go further in this direction and modify it a little bit and so on. But this is a little bit complicated. So luckily, the first equation here, you can solve it and see what is the calculated composition, put it in the inner loss function. And so we have just one loss function left, which you can derivate and solve. Now here we see some results. This is for a melanoma data set where we had about 3% of macrophages, and before training, that means all genes have the same weight. We see here the true cellular composition, and here's a predicted one, and this is a correlation between true and predicted, and this is about like 10%, so there's no prediction possible at all. But after gene weighting and selection, we get a nice correlation of more than 80%. So, okay, we can resolve for small cell populations accurately. The next problem we had, do we have when we have similar cell type profiles? Here we separated the T cells in 3 subpopulations, and we see especially for the 2 CD4 subpopulations, here and here, we have a very bad correlation, and after learning, we can improve for all. So we can also disentangle molecular related cell types. But now we are on our last problem. When you go back here, when we would just calculate the deconvolution on the main cell types, like with T cells, this sum of these 3 T cell subtypes would not necessarily be the same like we get on the main cell type. So it's it would be inconsistent. And therefore, we have to deal with inconsistent results when we're regarding different cell levels. And this we're regarding now. And here our idea was that we use an iterative top-down approach through our cell types hierarchy, so that we can use the results gained on the main cellular level and go down. This is a work I did with my master student, and this is a small part of the tree, like our real data which use the tree was way bigger, and we see here the major level in red, and for example the T cells are splitting up in CD4 and CD8 T cells. This is the minor level, which then split further up, the CD4 in T follicular helpers and regulatory T cells, which is in the subminor level. Sometimes we don't have a minor level, and it's going directly to the subminor level. And why do we want to do this? We want to use this hierarchy to predict small cell types, to improve the prediction of small cell types, and also to conserve the cell types higher up in the hierarchy, the results. So what is our pre-step? So pre-step, we're just concentrating on the main cell types, and this is we're using our digital tissue deconvolution algorithm, which I explained before, to calculate the cell type contributions of the main cell types and on normalized samples. And now we start our top-down approach. We say, okay. So first is, okay, I have here the T cells, and now I want to calculate the contributions of the CD4 and CD8 T cells. And the first step is okay, I create a residual box. It means I remove every contribution from the main cell types where I'm not actually interested in. So to say, I have the reference profiles, and I also have calculated how much they're contributing. So I can say, okay, I use the whole bulk and remove all the contributions of cells which are, for example, not T cells. Then in the next step, I again learn adjusted gene weights. And now I train them just on these T cell subtypes, because here I want to also algorithm to focus on genes which are separating these 2 subtypes the best. And the next step is, because I want to make sure that the child contributions are the same like the main contributions, I do normalization. First is, as we only regard the correlation in our loss function, we shift the results in a way that we get a line through the origins. This is the first line. We keep this, and then we make sure here that we say, okay, we normalize the contribution of this child terms to the parent term. And then when we go further, we would calculate residual bugs just for the CD4 T cells, and then adjust the gene weights of the T helper cells and regulatory T cells, normalize them. And this is we go through with this through every part of our structure. And the question is, how do we compete with other deconvolution methods? We competed ourselves with Base Prison, Saboside, and Music. And here I show exemplary for B cells and fibroblasts. When it's bold, it's the main cell type, and when it's italic, it's the sub-minor cell type level. And we see here, height has very high correlation values for all levels, and it's out-competing the other 3 methods. Here on the left, you cannot read this. This is just shown like this would be all cell types we were regarding, and in red is always marked which method had the highest prediction, and then we had the highest prediction except for one cell type. And very interesting is also what is the number of the sub-minor cell types with a correlation higher than 50%. So, because correlation higher than 0.5 means there is definitely a strong information confidence. And we see from the 34 sub-minor cell types, our algorithm had 31, which were above, and then the next one was Base Prison, which had just 22. And when we regard the mean over the prediction results, we see that here height had on all 3 levels, on the major, minor, and sub-minor levels, the highest correlation and was outperforming the other competing methods. And we were also regarding the normalized mean absolute errors. It means what is the percentage difference between the true value and the predicted one. So we want to have a low and my error, and we see that height has the lowest and my error for all competing methods. And what is also important, what I talked about, how to regard, how to calculate the contribution of the major cell types, or minor cell types, there are 2 ways. And we want now to check how consistent they are. First, you have the summing approach. That would mean I calculate all my cell type contributions on the sub-minor level and sum them up to the major level. And the other is that I can do an individual level approach, so to calculate all the levels independently. But then I do not hold the condition that a T cell is splitting up in all its child cell types, so that it can be a different result. And here, when I compare, I show, in the last line, I show you the correlation for the methods calculated with various approaches, and we see that the correlation is far from 1, and it should be 1 because normally you would expect that also child contributors are summing up to the main contributor. But this we can preserve because we keep this top-down approach. And after working on simulated bulk data, we now went to real bulk data from TCGA. We were regarding breast cancer subtypes, and we're calculating like the cell type contributions and now associating them with the survival. We had 4 subtypes, and all means, okay, we were now not regarding subtype, but just all bulks we had. And then we here see how much cell types are associated with survival. And when being associated with survival means we had a p-value below 0.05. And so we found candidates for all cell types. And now I'll show you an example. For example, we found that like the exhausted CD8 positive T cells, they were associated with improved overall survival. So we had to find something in the literature which is confirming our findings, and we found that high levels of this exhausted CD8 positive T cells promote the formation of tertiary lymphoid structures, and these tertiary lymphoid structures facilitate antigen presentation and lymphoid cell recruitment within the tumors. So, okay, this makes sense. And also these exhausted CD8 positive T cells, they help for interaction with immune therapies and can enhance the therapeutic responses. Okay, so literature and our findings are the same. And for different T cell subgroups, regulatory T cells, we found that they are associated with a poor prognosis in triple-negative breast cancer. And literature says that this regulatory T cells can suppress the immune activity and thereby impairing the immune response against the cancer cells. And though one see why it is important to know, to have information about the cell type contributions on the subminor level, because would we only regard the T cells, we would have found nothing significant. But when we go further down, we see that the T cells are very important to predict if it's a good or a bad outcome for the patients. So to sum up what we can estimate the minor cell populations, we can disentangle molecular related cell types, and we get consistent results when regarding different cell types levels. And this is important because there are 2 points. When we have the further down you go, the more inaccurate your results go. So when you calculate on the lowest level and send them up, you would take errors from the lowest level and bring them right to the top where you could predict more accurate. And also when you go down and you see, okay, now on the minor level, here the results are inaccurate. I would not go down further and split it further out because it seems our data is not providing the accuracy to predict correct on a level further down. So here are the availability. We have our code available as a Python implementation. Our paper is published now via the conference, and we also have this first algorithm, this DTT algorithm, available as a graphical user interface, and we will implement our height algorithm in the next time also in this Deconomics user interface. I want to thank my collaborators from the University of Bergen, the University Hospital, the University Medical Center in Göttingen, and the Medical Center in Hanover, and my PhD student from Regensburg, Dennis Völkel.
Questions?
Hi, thank you very much for a very interesting talk. So I'm not sure I understood, when do you decide to stop the deconvolution, not go further down? Is it because you can decide that those subtypes are not present in the sample, or why would you stop?
I would stop when I see my correlation is, for example, just 0.2, then I say, okay, I cannot go further down because I know I cannot predict correct on a level higher above in the hierarchy. So when I have here a bad result, because I have not enough marker genes or the data is not available so that I can go further down, why should I go further down when I know the major cell type is badly predicted?
So how are the marker genes? They're not expressed, or why would
no, it just can be that they're not expressed. It can be that there is not enough of the cell type at all in the mixtures. This could be biological reasons.
Thank you. This is very interesting because we did this population from single cell data in triple negative, and we see 100 sub-minor cell types. Do you think you could do that on the TCJ TNBC?
I don't know if we can go so deep. The point is for us, when we have the training data, where we need single cell data with labelings, then we can go further and further down. But the further you down, the more similar
subtypes.
The subtypes are getting, and also the smaller the proportional, so the more inaccurate you get. But in theory, there's no stop.
Thank you very much. That was a really nice talk. I was just wondering, in real deconvolution, you often have this technical mismatch. So your single cell will have either a strong 5' or 3' bias, but your bulk RNA will capture the whole transcript. Have you thought about trying to include that in the model or learn it from the data?
Yes, we thought about it, and we at the moment we're doing this, that we are we have, for the TCGA, we also had some normal samples, so we could say, okay, what is a fold change between single cell and bulk? So you could like recalculate the domain shift for the cancer response. But yes, we are also actually now working it and implementing this computationally, and we have at the moment also quite promising results, and we're testing this and want to publish this in 3 months, half a year. Yeah, we thought about it and we're working on it.
Hi, nice work. I guess it's a very similar question, but because all the assessment is done on pseudobulks, if I understand.
No, we did the training and the testing on pseudobulks because we need the pseudobulks that we create for the training enough power to do learning on it. And for testing, it's important that we can control each other. But this is what I showed you on the breast cancer. This is a normal bulk from TCGA.
Breast cancer, I understand, but the performance values.
Yes, because I can only validate me when I have a ground truth, which I.
Yeah, no. So I was wondering if you had tried to use FACS data.
Yes, we did this during my PhD. We had a sample where we had FACS also, and the results were good. Like we had some 18% also correlation, like.
Okay, let's thank the speaker again.